Okay, next thing we want to consider is this idea of membrane polarity. Okay, I've talked a little bit about the fact that the plasma membrane is a hydrophobic barrier, so it's a good insulator. And if we utilize it to separate charge, it's going to be extremely difficult for opposite charges to come into contact with each other because the lipid portion of the phospholipid bilayer has insulating qualities very similar to, say, rubber or plastics that you see as insulators in common devices that use electrical power. There's a separation of charge across the plasma membrane. There's a thin layer of positive charge outside the membrane and a thin layer of negative charge inside the membrane that are separated by the phospholipid bilayer. This electrical gradient provides energy for the cell to do work. Electrical potential found across the plasma membrane is called membrane potential, and the study of how these potentials relate to living processes is called electrophysiology. Resting membrane potential is the membrane potential that occurs when the cell is at rest, and it's measured in millivolts. The value is negative, meaning that the inside of the cell is more negative than the surrounding extracellular fluid. Now, you might say, okay, well, how is this resting membrane potential generated? And we have three factors that are critical to understand in the generation of membrane potential. The first factor is the fact that there are leak channels for sodium and potassium cations embedded in the membrane. So leak channels are defined as channels that are always open. Okay. Number two, okay, the sodium-potassium pump maintains a separation of charge across the plasma membrane by pumping 3 sodiums out for every 2 potassiums that flow in. And then number 3, important factor, is the insulating quality. I'll just write insulation here. Of the phospholipid bilayer itself. Okay. So what we find in the plasma membrane is that there are more potassium leak channels than there are sodium leak channels. So potassium tends to pass from the cytosol to the extracellular fluid more quickly than sodium tends to flow in the opposite direction. But the pump compensates for this leakage and helps to maintain this separation of charge, which can be harnessed just like separation of charge in a battery to perform cellular work. Another type of active transport is through vesicles. A vesicle is simply a bubble, in a sense, inside the cell that is bordered by phospholipid bilayer and is used as a vehicle to carry substances that are manufactured into the cell to the cell membrane, where they can fuse and release their contents, and that's a process called exocytosis. Or we can also perform the opposite function, where we can cause a portion of the plasma membrane to invaginate, bringing a substance inside the cell, and that's a process called endocytosis. But this all requires energy because it won't happen spontaneously. This active transport uses carrier proteins and channels that are effective, but the vesicle transport kicks in when the macromolecules are too large to fit through channel proteins in the membrane. So we have to have some other way of getting them across the cell membrane, and that's where the vesicles come in. So vesicles are small sacs that are filled with large molecules too big to be transported by other means across the cell membrane. They're enclosed in a phospholipid bilayer that allows them to fuse with or be formed from the plasma membrane or other membrane-bound organelles. It's an active transport process. It requires energy from ATP in order to move these vesicles from one place to another. Okay, so bottom line, we have to have this because some stuff that the cell makes and spits out and some stuff that the cell needs to absorb are too large to fit through a single gated or ungated channel or through a pump. Okay, it's just the size consideration is something that we have to consider here. So let's take a look at vesicular transport.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
In endocytosis, we're bringing substances into the cell from the external environment. Receptor-mediated endocytosis is similar to pinocytosis in that it uses receptors to fill vesicles with a specific molecule. And so you can see here that there's an area of the membrane where we have a lot of receptors. These are frequently transmembrane. Okay, they go span the entire plasma membrane. The binding site is on the extracellular matrix side of the plasma membrane. The ligand attaches to these receptors, and then this triggers invagination. Okay, normally in receptor-mediated endocytosis, there are proteins that also often stabilize this vesicle as it pinches off from the plasma membrane. In exocytosis, we have a large molecule that's manufactured inside the cell that will exit the cell, and this is what we also know as secretion. The vesicles will fuse with the plasma membrane and open into the extracellular fluid, releasing their contents, while transcytosis describes molecules that are brought into the cell by endocytosis, are transported by the cell to the opposite side of the cell, say from the apical end of the cell to the basal end, and then secreted by exocytosis across the basal end. And all of this movement, as you can imagine, is going to require cytoskeletal elements, motor proteins, and the use of ATP as a fuel source, because none of this would happen spontaneously. So let's take a look at membrane transport.
In order for us to use the molecules and energy we get from food, we must transport them across cell membranes. Transport of nutrients is essential for all cells, but we'll use cells in the small intestine to teach the principles of membrane transport. Let's zoom into the surface of an intestinal cell. The plasma membrane is selectively permeable, Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can move freely across the lipid bilayer. Notice that the lipid molecules move down their concentration gradient from where they are more concentrated to where they are less concentrated. This is an example of simple diffusion. Diffusion is a form of passive transport. It does not require energy from the cell, but it does depend on the concentration gradient, size and charge of the molecule or ion. However, most molecules cannot cross the lipid bilayer. Instead, molecules like the sugar fructose move into intestinal cells by facilitated diffusion, moving down their concentration gradient and through transport proteins. It also differs because it acts only on specific molecules, and the transport proteins can become saturated, reducing the amount of diffusion. However, facilitated diffusion doesn't require energy from the cell, so, like simple diffusion, it's a form of passive transport. The majority of water molecules cross the plasma membrane by facilitated diffusion, also called osmosis, through specific protein channels called aquaporins. Some ions, such as sodium and potassium, require transport against their concentration gradients. The sodium-potassium pump actively transports these ions from where they are less concentrated to where they are more concentrated. This process requires energy from the cell and is known as active transport. Energy from ATP is used to move 3 sodium ions, shown in red, out of the cell, and 2 potassium ions, shown in blue, into the cell. Because the pump uses ATP directly, this type of transport is called primary active transport. Another type of active transport, secondary active transport, also moves chemicals against gradients. It doesn't use ATP as a direct source of energy. Instead, it uses the downhill gradient from one ion or molecule to drive the uphill movement of another ion or molecule. For example, sodium and glucose move into the cell by secondary active transport in a process called cotransport or symport, because both substances are moving in the same direction. Sodium ions move down their concentration gradient created by the sodium-potassium pump, but because glucose concentrations inside the cell can quickly exceed those outside the cell, glucose moves against its concentration gradient. The cell moves larger materials, such as proteins, from the intracellular fluid to the extracellular fluid, using vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. Cells use all these processes, passive and active transport, exocytosis and endocytosis, to move materials across their membranes.
Okay, our next topic is cytoplasmic organelles. Now, bottom line, if we look at our model of the cell, what is an organelle? It's the cellular machinery that carries out the life process of the cell. Usually these chambers inside the cell, or these non-membranous organelles, have a specific task that are vital to maintaining homeostasis. Some are separated from the cytosol by a membrane, which we call compartmentalization, while others are not enclosed in a membrane. Membrane-bound organelles include mitochondria, peroxisomes, endoplasmic reticulum, both smooth and rough, the Golgi apparatus, and lysosomes, and they perform functions that could be destructive to the rest of the cell, Organelles that are not enclosed in a membrane include ribosomes and centrosomes. Now, what you'll notice, right, looking at this model of the cell, is that we have these different chambers. Okay, mitochondria. We have rough and smooth endoplasmic reticulum. We have Golgi apparatus. We have lysosomes and peroxisomes. We have inclusions. Okay, we have a nucleus. We have a nucleolus, and we have centrosomes. All of these different chambers, okay, are engaging in some sort of cellular process. The purpose of containing it in a chamber is so that the process is more efficient. It also aids in separating chemical reactions that would work at cross purposes to each other. For instance, if we had an enzyme whose job it was to tear up a particular molecule, let's diagram this. Let's say that we had an enzyme whose job it was to take molecule AB, okay, and to crack it into molecules A and B, okay, and we would call this enzyme number 1. And let's say that we had another enzyme whose job it was to catalyze the reverse reaction, okay, where we take A and B and we assemble them in order to make molecule AB. Okay, this would be enzyme number 2. If enzyme number 1 and enzyme number 2 were in the same physical space with their substrates, we would engage in what's called a futile cycle. Where we would continuously assemble and disassemble the same molecule, burning energy and creating heat, but not really accomplishing anything for the cell. And thus, we have to separate these reactions that are polar opposites in order for the cell to function as a living unit. Mitochondria are described as the power plant of the cell. These are membrane-bound organelles involved in chemical energy production, and they provide most of the ATP used in the cell. Each mitochondria has its own DNA, enzymes, and ribosomes, which are, we're going to find out, those are organelles that are involved in protein synthesis. The membrane is a double bilayer. It has a smooth outer membrane, and the inner membrane is highly folded into these little ridges and grooves known as cristae. Each membrane has its own unique enzymes and proteins required for specific functions. The outer membrane contains large channels that allow molecules from the cytosol to enter the inner membrane space between the two phospholipid bilayers, while the inner membrane is selective and transports only necessary solutes into the matrix using specific transport proteins. The matrix contains mitochondrial DNA, proteins and enzymes specific for the breakdown of organic fuels by oxidative catabolism, okay, and also contains the DNA of the mitochondria and the ribosomes of the mitochondria. Now you might be thinking, why in the world does the mitochondria contain DNA? We are familiar with DNA as the genetic material, basically the protein building instructions for the cell. So why does a mitochondria have DNA? And the answer is that we believe that the mitochondria is an obligate endosymbiont that is a descendant of a very primitive bacteria. And the reason we believe this is because when we compare mitochondrial DNA to bacterial DNA, we see that there's a very close match in DNA sequence, in addition to the fact that the ribosomes that mitochondria use resemble bacterial ribosomes more so than the ribosomes that we expect to find in the cytoplasm of a cell in our own tissues. So the theory goes that early on in evolution, a relationship was fostered in which these primitive bacteria would live inside a primitive version of a eukaryotic cell. A eukaryotic cell is defined as one that has a membrane-bound nucleus and membranous organelles, with the idea that the eukaryotic cell would provide a home and a constant source of fuel for the primitive bacteria, while the primitive bacteria would provide all the energy necessary both to maintain itself and the eukaryotic cell. And over time, these two entities became so dependent on each other that they were unable to exist independently, the result being that eventually they lost their ability to live outside the confines of a eukaryotic cell. Okay? Another example of an organelle that we believe was derived in the same fashion is the chloroplast, which we find inside of plant cells, which is designed to take the radiant energy from the sun, the UV radiation, and transfer that energy into the manufacture of sugars, such as complex carbohydrates like starch and cellulose, which provide structure and fuel for the plant, and as a result allow the plant to take advantage of a job in the ecosystem that no other organism could effectively fulfill, the job of converting UV energy from the sun into chemical potential energy that can be used by not only the plant, but species that consume the plant material. So very interesting etiology here with the mitochondria. Now you might wonder, okay, why these cristae in the intermitochondrial membrane? And anytime you see an unusual surface feature, in this case grooves and ridges, always think about surface area. And the idea behind these cristae is that they add surface area to the intermitochondrial membrane that can be used for the manufacture of ATP in a minimal volume. Yeah, we're going to see the same motif repeated in organs such as the brain and the digestive system. Here we see it in an organelle. Okay, our next organelle is the peroxisome. These are membrane-bound organelles that use oxygen to carry out several chemical reactions that generate hydrogen peroxide, which oxidizes toxic chemicals to less toxic compounds that can be eliminated from the body before they cause damage. Catabolic reactions in the peroxisome break down fatty acids into smaller molecules that can be used for energy production and other anabolic reactions. Certain phospholipids synthesized in peroxisomes are critical to plasma membrane of specific cells or in the nervous system. Ribosomes are protein synthesis organelles. These are tiny granular non-membrane bound organelles that manufacture proteins by assembling amino acids into a chain, which we know from the previous chapter is the primary structure. It's made up of a large and small subunit. Each is made up of ribosomal proteins and ribosomal RNA. Some are found free in the cytosol and make proteins needed within the cell itself, while others are bound to the membranes of other cellular structures, primarily the rough endoplasmic reticulum, and generate proteins destined for export outside the cell, for export to a lysosome, or for insertion into the membrane of a membranous organelle or the plasma membrane, or say the nuclear membrane, for instance. The endomembrane system describes a series of vesicles that exchange proteins and other molecules. They synthesize, modify, and package these molecules and then transport them to the area in the cell where they're going to perform their task. The plasma membrane, the nuclear envelope, and the following organelles are all components of this system. It includes the endoplasmic reticulum, which is made up of the rough endoplasmic reticulum, or RER, and the smooth endoplasmic reticulum, or SER, the Golgi apparatus, and the lysosomes. The endoplasmic reticulum is a large folded phospholipid bilayer continuous with the nuclear envelope and exists in two forms, the rough and the smooth ER. The rough ER has ribosomes attached to it, while the smooth does not. The rough endoplasmic reticulum, with its bound ribosomes, has protein synthesis going on at its surface. The products enter the rough endoplasmic reticulum lumen from the ribosome. Incorrectly folded polypeptide chains are detected and sent to the cytosol for recycling. Most proteins that enter the rough endoplasmic reticulum are destined for transport out of the cell. It also packages secretory proteins into transport vesicles made of a phospholipid bilayer that are sent to the Golgi apparatus to be processed and labeled. It produces membrane components for membrane-bound organelles in the plasma membrane, including integral and peripheral proteins. While the smooth endoplasmic reticulum is not ribosome-associated and essentially has no role in protein synthesis, but it stores calcium by pumping calcium out of the cytosol for future use. It's capable of detoxification, limiting damage caused by certain substances such as free radicals, which are electron-poor species that essentially steal electrons from the chemical bonds of functioning molecules, destroying them. And it's also involved in lipid synthesis by manufacturing the majority of plasma membrane phospholipids and cholesterol, as well as a number of lipoproteins and steroid hormones. The Golgi apparatus is located between the rough ER and the plasma membrane, and it's a group of flattened membranous sacs filled with the enzymes that are required to label the proteins that come into it from the rough ER, as well as other molecules. Proteins and lipids made by the endoplasmic reticulum are modified, sorted, and packaged for export by the Golgi apparatus. Products packaged in the Golgi can be secreted from the cell by exocytosis or become part of the plasma membrane or be sent to the lysosomes for recycling. So what do we have here in a sense? Let's sort of diagram this. If we start out with a messenger RNA, okay, we know that the messenger RNA is going to be used by the ribosomes to make protein. Okay. Let's consider that this is a membrane-bound ribosome. So it's associated with the rough ER. This protein gets extruded inside the rough endoplasmic reticulum, where it will begin to fold into its native conformation. And remember that what drives that is the primary amino acid sequence, causing this protein to adopt the shape that is essentially the low energy state for that particular polypeptide, and also the state in which that protein is going to perform its final function inside the cell. It'll work its way through the rough ER and eventually be loaded into a vesicle, which will fuse with the Golgi apparatus, which will tag the protein as to its destination, make what we call post-translational modifications, to the protein. And then, based on the chemical tags that are attached to the protein, the Golgi will load these tagged proteins into secretory vesicles, where they will be transported based on the identity of the secretory vesicle to the plasma membrane or other organelles. Okay, and then in some cases they'll be transported to lysosomes, okay, which are a type of membrane-bound organelle primarily tasked with destruction of stuff that's no longer useful. Okay, so what we have here is a system. You could think of the ribosomes as the workers in the factory that assemble the product. Okay, the protein is the product. The rough endoplasmic reticulum you could think of as the product being loaded onto a vehicle and transported to the major distribution warehouse. Okay, the vesicles bring the product to the warehouse. There could be the trucks driving from the warehouse to the distribution center. Okay, and the Golgi is the distribution center for the product, which is then loaded onto a different set of trucks and then driven off to the stores where it's going to be sold. Okay, so it's very much like that. Okay, we manufacture the product by the ribosomes extruding the protein into the ER. The protein folds up based on energy. It gets loaded into a vesicle. The vesicle fuses with the Golgi. The Golgi tags it, modifies it, loads it into a secretory vesicle, and then off we go to our final destination. Now, as you can imagine, any error in this transport process and distribution process could have disastrous consequences for the cell. In cystic fibrosis, some cells are missing a protein component of the chloride ion channel, which causes deficient chloride ion transport in the lungs and the digestive and integumentary systems, and results in abnormally thick mucus that blocks airways, causes digestive enzyme deficiency, and generates a salty sweat. This is the result of a mutation that causes the chloride channel protein to misfold in the rough ER. The protein is therefore destroyed, even though it would be functional if it were inserted into the membrane. In short, this disease is caused by an overprotective rough endoplasmic reticulum that's destroying something that would normally be useful. Now, another example of a condition in which we compromise cell function as a result of a defect in the manufacture and distribution of protein is mad cow disease. In mad cow disease, what happens is that proteins that are critical for the function of nerve cells misfold in the rough endoplasmic reticulum. Those misfolded proteins are actually what are called chaperones. A chaperone is basically a protein that helps other proteins properly fold. The defective chaperones misfold other proteins, resulting eventually in the destruction of the entire cell. The result in mad cow disease is that we see a degradation of nerve tissue, a loss of function of the central nervous system, and death usually within a few days. And this is thought to result from the practice of including bone meal in the feed of cattle. The bone meal contains nervous tissue, and the nervous tissue that's ingested across the gut and into the bloodstream contains the prions, the proteinaceous infectious units, the renegade chaperones that misfold the protein in the nervous tissue. Okay, the endomembrane system, as we've discussed, is a mechanism to manufacture and distribute proteins to areas required in the cell for those proteins to do their job. So essentially a way to get the proteins that enable the cell to remain a living unit viable by delivering them to the place where they're going to perform their task. Lysosomes are organelles responsible for the digestion of worn-out cell components or misfolded proteins, or in some cases, when the cell has outlived its usefulness, it leads to the destruction of the entire cell. It contains digestive enzymes called acid hydrolases. In this special chamber, the lysosome, macromolecules are broken down into small subunits. They can be released to the cytosol for disposal or are used to manufacture new macromolecules. So I always like to nickname the lysosome the stomach of the cell. That's one of the ways that I remember its task most easily, right? The stomach of the cell. Now, as you can imagine, if we have a problem with the lysosome, we have a problem with the proper disposal of cellular components that need to be recycled or degraded in order for the cell to continue to function. And so there are a whole host of diseases called lysosomal storage diseases in which molecules that would normally be broken down by lysosomes fail to do so, and the result is that those products accumulate inside the cell. A perfect example is a condition known as Tay-Sachs disease. And in this genetic condition, this is a recessive disorder. It's autosomal. Okay. We fail to break down a certain class of molecule called a ganglioside, and this results in degeneration of nervous tissue and usually death in infancy. And it's something that we can screen for using amniocentesis or CVS. So this is a summary of the functioning of the endomembrane system. We kind of diagrammed it earlier, but let's go through it in detail. The events that ensue from the production of a messenger RNA to the delivery of that protein product to the point in the cell where it's going to perform its task starts with the smooth endoplasmic reticulum manufacturing lipids and the rough endoplasmic reticulum producing proteins, and each product is packaged into vesicles for transport to the Golgi. The Golgi then sorts and modifies both lipids and proteins and packages them into vesicles, which may take 3 pathways once they exit the Golgi. The vesicles may be sent to the lysosomes where they undergo catabolic reactions, basically breakdown. The vesicles may be incorporated into the plasma membrane or the membrane of another organelle in the cell, or the vesicles may be sent to the plasma membrane where they are secreted by exocytosis out of the cell. And so you can see the entire diagram of the route from manufacture to distribution here. A group of diseases that results from the deficiency of one or more acid hydrolases in lysosomes are classed as lysosomal storage diseases. Examples include Gaucher's disease, where a deficiency of a particular enzyme causes the accumulation of glycolipid in the blood, spleen, liver, lungs, bone, and sometimes the brain. Its most severe form is fatal in infancy or early childhood. And then Tay-Sachs, which, as we already detailed, results in the accumulation of glycolipids in the brain. This leads to progressive neural dysfunction and death, usually around age 4 or 5. Okay, our next topic is the cytoskeleton, which is made up of several types of protein filaments. It's a dynamic structure that can change the function of, they can change its function based on the cell's needs, and it plays a variety of roles in normal cell function. It gives the cell its shape and size by creating a framework. It provides strength, structural integrity, and anchoring sites for the support of plasma and nuclear membranes and organelles. And it allows for cellular movement, where protein filaments are associated with motor proteins, which can harness the chemical potential energy of ATP to turn that into kinetic energy and move components inside the cell from one place to another. And it performs specialized functions in different cell types, for example, phagocytosis by macrophages or contraction by muscle cells. Okay, so let's sum it up. Okay, the cytoskeleton gives the cell shape. It allows the cell to move cellular components around inside the cell, and it allows for endocytosis and exocytosis. And also, we're going to find out, plays a critical role when the cell gets ready to divide in the process known as mitosis. The cytoskeleton has 3 types of long protein filaments composed of smaller protein subunits that allow for rapid disassembly and reassembly. These include actin filaments, intermediate filaments, and microtubules. Actin filaments are the thinnest filament and composed of intertwining strands of actin subunits and provide structural support, bear tension, and maintain the cell shape. They're involved in cellular motion when combined with myosin. Intermediate filaments are rope-like assemblies made up of fibrous proteins such as keratin. These are more permanent structures, and they form the framework of the cell and anchor organelles in place. and help organelles in the nucleus maintain their shape and size, and help cells withstand mechanical stress. And finally, microtubules are the largest filaments. These are hollow rods or tubes made up of alpha and beta tubulin that can be rapidly assembled and disassembled at a special organelle known as the centrosome. They maintain the internal architecture of the cell and they keep the organelles in alignment. They also interact with the motor proteins dynein and kinesin that allow for vesicles to be transported along the microtubule network from one place to another. Microtubules extend out from the centrosome, which consists of a gel matrix containing tubulin subunits. When the cell is not dividing, the centrosome is a microtubule organization center located near the nucleus. A pair of centrioles composed of a ring of 9 groups of 3 modified microtubules is critical for cell division. The basal bodies are modified microtubules that are found on internal surfaces of the plasma membrane where flagella and cilia originate. Now, flagella are used for locomotion, and cilia are used to propel substances across the apical surface of the cell from point A to point B. So, bottom line, what does the centrosome do? It assembles and disassembles microtubules as needed, okay, and also acts as sort of the reel on a rod and reel. It's able to pull on these microtubules in order to move cellular components around. One of the most obvious examples is the movement of chromosomes in mitosis and meiosis, which is powered by the actions that take place within the centrosome. Cellular extensions are formed by the inner framework of the cytoskeleton. These include microvilli, cilia, and flagella. Microvilli are finger-like extensions of the plasma membrane that contain actin cores to maintain shape. And these are examples of the structure-function core principle. Again, whenever you see an unusual surface feature, you should always think first of surface area. And what this does is it increases the surface area of the apical end of the cell so that secretion and absorption are more effective within a minimal volume. Cilia are hair-like projections composed of microtubules and motor proteins that move in unison to propel substances past the cell. Okay, a perfect example is the epithelia that line the inside of the upper respiratory system. The cells that produce mucus, called goblet cells, extrude the mucus into the lumen of the trach. The mucus traps particulates in the air that we breathe, and the cilia that are on the apical surface of the epithelial cells move that particulate-laden mucus up towards the oral cavity, where we can either spit it out or swallow it. In any case, it does not end up in the lungs, where it would produce damage to the delicate epithelial tissue of the lungs, causing them to scar. They're found in great numbers on the apical ends of the cell normally. Flagella are solitary and longer than cilia. Their only location in human cells are on sperm cells, the male gamete. They beat in a whip-like fashion and propel the entire cell from point A to point B. Flagella and cilia both are structurally similar to centrioles, except they contain 2 central microtubules not found in centrioles. Okay, so cellular extensions, right? Microvilli for increasing surface area, cilia for the propulsion of components across the cell surface, and flagella for locomotion. Of these, cilia and flagella have a microtubule structural basis. Okay, our next topic is the nucleus. It's the governing body that directs the activities of other cellular components, and it determines the type of proteins and the rate at which the cell makes them. DNA is housed in the nucleus and contains the code for nearly every protein in the body. These plans, which are arranged in information blocks called genes within the DNA, are excluded by several types are executed by several types of RNA which build a wide variety of proteins. The nucleus consists of three main structures: the nuclear envelope, which is a double-layered membrane that surrounds the nucleoplasm, which is basically cytosol that contains water, free nucleotides, enzymes, as well as proteins that are associated with the DNA and RNA, and of course the DNA, which is the genetic material, and the RNA, which is found in several different places: messenger RNA, which essentially are copies of the genetic information that are used by ribosomes to generate the proteins; transfer RNAs, which serve as adapter molecules that bring amino acids to the site of translation so that we can build the protein from its building blocks; and ribosomal RNA, which is a structural component of ribosomes, which are the protein synthesis machinery in the cell. DNA and associated proteins are found in the nucleoplasm as a loose structural arrangement known as chromatin. One or more nucleoli are found suspended in the nucleoplasm. Now, a couple of words about chromatin and nucleoli. Okay, let's do this in blue. The DNA double helix, which I'll show here as just a blue filament. Okay, if it were just naked inside the cytosol of the nucleus, could easily be damaged as a result of shear force. Okay, and if it were in this extremely extended conformation, it would take up a very large volume. So one of the ways that we try to protect the genetic information and compress it into a smaller volume is to wind it around special proteins called histones. Okay, so I'll draw you here some histones in green. Okay, histones have a roughly globular shape. They're packed in an arrangement similar to this. And what we do with the DNA is we wind it around these histones in order to protect it and to reduce the volume that it takes up inside the nucleus. So if we have here a strand of DNA coming in, we would wind around the histone 2.5 turns, okay, and then we would extend out to the next block of histones and continue the process. Okay, so here we've got a few more histones here, okay, and we wind around it again 2.5 turns, and then the motif continues. Okay, and so what this does is it reduces the volume of DNA and protects it. Okay, and it's also thought that the histones may interact with other proteins that control transcription. Okay, the nucleolus, which is an area in the nucleus that stains a little bit darker than the rest of the chromatin, you can see it in here, is the site of ribosome assembly. Basically, these are locations of the transcription of genes for ribosomal RNA, which become a structural component of the ribosome, which then is allowed to exit the nucleus through the nuclear pores and then assume its position either on the rough ER or in the cytoplasm. The nuclear envelope is a double phospholipid bilayer similar to that we see in the mitochondria, and it surrounds and encloses the contents of the nucleus. The outer membrane has ribosomes that are attached to the surface, and it's continuous with the rough ER. And the inner membrane lines the interior of the nucleus and is supported by a network of intermediate filaments known as the nuclear lamina. Nuclear pores are found where the outer and the inner envelope membranes come into contact, and they serve to connect the nucleoplasm with the cytoplasm and allows for substances to move between the two locations. Remember that um, in order to generate a transcript, one of the things that we have to do, for instance, is manufacture the protein known as RNA polymerase, which is responsible for synthesizing the messenger RNA from the genes that are contained in the nucleus. Well, RNA polymerase would be too large to pass through this double-layered membrane on its own. It needs a hole to go through, and that's part of what the nuclear pore provides. Okay, so these are examples of very, very large holes compared to normally what we see with channel proteins such as leak channels and pumps and all that we see in the plasma membrane. So this allows material, large material, to go from one side of the nuclear membrane to the other without having to expend a lot of energy. Chromatin consists of one extremely long DNA molecule and its associated proteins. These organize and fold the molecule to conserve space but also to protect the information. A nucleosome is defined as a strand of DNA coiled around a group of histone proteins and appears like beads on a string, and it reduces the length of the strand by approximately 1/3. But as we'll see, there are further layers of condensation that reduce the volume of DNA and protect the information. During periods of cell division, chromatin threads coil tightly and condense into thick structures known as chromosomes. The human cell contains 23 pairs of chromosomes, one from the father and one from the mother. Sister chromatids are identical copies of each chromosome, and they're made in preparation for cell division and connected to each other at a region known as the centromere. Okay, so when a cell gets ready to divide, a few things happen. In preparation for mitosis, we synthesize a new copy of the DNA that's contained in the nucleus through semi-conservative replication, where one old strand is used as the template for the new strand. And then we condense that genetic material into visible chromosomes, as you can see here in the diagram. And the reason being that if we're going to be pushing and pulling this genetic material around the cell, we don't want it to be damaged. And so in the chromosome configuration, it's less likely to destroy the genetic information than if it were the open chromatin, relatively open chromatin that we see in the nucleus when the cell is engaging in its normal task in the body, which we know as interphase. Okay? So it's a way of basically boxing up your information so that you don't wreck it before you distribute it to the two new cells.